Hello, my name is Noreen. Welcome to my science flash on a comparative analysis of the nutrigenomic effect of L-carnitine on the transcriptional regulation in liver cells and adipocytes under hyperglycemic and hyperlipidemic growth conditions. Our supplement of interest is the nutrigenomic factor L-carnitine. But where do we get it from? The largest amount is obtained through carnitine-containing foods, with meat being the main source. Besides the nutritional supply, our endogenous biosynthesis accounts for about 25% of our L-carnitine requirements. It has multiple biological functions. The most important one for this science flash is the involvement of carnitine in the energy metabolism, as it is crucial for the transport of long-chain fatty acids from the cytosol into the mitochondrial matrix, and thereby carnitine plays a pivotal role for the energy supply through beta oxidation. This slide shows you the procedure, how we performed our cell culture study. First of all, cells were grown in normoglycemic and hyperglycemic media and were then supplemented with different carnitine concentrations. Lipid supplementation was used to simulate hyperlipidemia, and our control, so to say the healthy patients, are the cells that were grown under normoglycemic conditions and without any carnitine supplementation and not being dialyzed. Finally, we focused our interest on the genes that you can see in the lower box. Let's now take a look at the investigated genes and how their expression rates are influenced by the different metabolic conditions. Let's start with the glucose transporter GLUT2 that is typically located in fat and muscle cells and is responsible for the removal of glucose from the circulation. What we see very nicely here is that GLUT2 gene expression strongly depends on the glucose levels in the cell, with expression rates being manifold increased under hyperglycemic conditions compared to cells that were grown in normoglycemic medium. This is logical, as the higher glucose concentration affords higher glucose shuttling into the cell. The use of dialyzed FCS, so no carnitine in the medium, has a massive effect on the gene expression. We observe a drop under hyperglycemia and an induction of the gene expression under normoglycemic condition, which shows that carnitine is involved in the gene regulation of GLUT2 transcription. Let's zoom in a little bit to see better the effect of carnitine supplementation. If carnitine is added, we observe a reinduction of the GLUT2 gene expression under hyperglycemic conditions. This proves that carnitine is an important nutrigenomic factor for the expression of GLUT2 and has an anti-diabetic effect, as it enables more glucose uptake into the cell by increasing the gene expression. This effect is only observed if carnitine is supplemented in physiological concentrations of 40 micromolar. Higher carnitine concentrations induce the opposite effect, and the gene expression is repressed again, but still remains on a higher level compared to the normoglycemic control. Under normoglycemic conditions, we observe total, distinct, and opposite effects of carnitine supplementation. Physiological carnitine supplementation represses the GLUT2 gene expression, whereas higher carnitine concentrations lead to an increase of the gene expression. For PPAR-gamma, a nuclear receptor that is mainly responsible for energy storage via adipogenesis, we see a similar pattern as for GLUT2. Again, we observe different gene expression rates depending on the glucose levels. We also observe carnitine concentration-specific changes that depend on the certain growth conditions. We see either a reinduction or repression of the gene expression. The same pattern is also observed for the CRUT gene that plays an important role in the energy homeostasis. Now we are coming to the gene BCOX, which is highly expressed in breast carcinoma. Here, I would like to put your focus on the carnitine concentration-specific influence on the gene expression. You can see that carnitine induces the gene expression under normoglycemic conditions, but apparently it represses it to extremely low levels if it is supplemented in supraphysiological concentrations. This shows that the concentration of carnitine is very, very crucial and can lead to reversed effects. Under hyperglycemic conditions, however, we observe opposite effects, and interestingly, the highest carnitine concentration of 120 micromolar puts the expression rates to almost physiological levels. We observe transcriptional regulation through glucose and carnitine also in our liver cell line. Especially distinct is that high glucose levels lead to a significantly higher gene expression. Again, under dialysis, we observe a strong downregulation. Taking a closer look to GLUT2 expression under normal glycemic conditions, we see that carnitine is able to induce the gene expression significantly. We conclude that carnitine modulates the transcription levels of all investigated genes, which again emphasizes its function as nutrigenomic factor. Firstly, we observe that the gene expression rates strongly depend on the glucose levels in the cells. Furthermore, we have seen that the carnitine supplementation had opposite effects on the gene expression depending on the glucose concentrations. We also conclude that the gene expression rates depend on the specific carnitine concentration. In most genes, we observed something like a cross point or a critical concentration, where the opposite effects occurred as a certain carnitine concentration is added. Last but not least, we observed that also the lipid supplementation had a significant influence on the gene expression rates of the investigated genes. As a take-home message, I would like to emphasize the involvement of carnitine on the regulation of genes that are very important for the glucose and lipid metabolism. Taking into account its anti-diabetic effect, it might be especially interesting for patients with type 2 diabetes to stick to a diet rich in carnitine. Thank you for your attention.